Niacin's emerging role in cellular cholesterol management offers intriguing therapeutic possibilities beyond cardiovascular disease, particularly in the context of lysosomal storage disorders like Niemann-Pick type C, (NPC). At the heart of NPC pathology lies a defect in the Niemann-Pick C1, NPC1 protein, which is essential for the transport of cholesterol out of the lysosome. In its absence or dysfunction, cholesterol becomes trapped within these organelles, leading to widespread lipid accumulation, cellular toxicity, and progressive neurodegeneration. While current treatment options for NPC are limited and largely supportive, the finding that niacin upregulates NPC1 expression introduces a potentially novel avenue for intervention. In macrophages loaded with oxidized LDL, niacin has been shown to significantly enhance NPC1 protein expression. This effect is driven by a well-defined signaling cascade. Niacin activates CD38, an enzyme that catalyzes the production of NADP, a potent calcium-mobilizing molecule. NAP, in turn, triggers calcium release from lysosomes through TRPML1 channels, which promotes the activation of liver X receptor alpha, LXRA. LXRA is a nuclear receptor known to transcriptionally regulate genes involved in lipid metabolism, including NPC1. The result is a marked upregulation of NPC1, leading to enhanced cholesterol efflux from lysosomes and improved lipid trafficking within the cell. This mechanism has profound implications for disorders like Niemann-Pick type C, in which NPC1 function is partially impaired rather than entirely absent, as seen in many missense mutations. In such cases, increasing the expression of residual NPC1 protein or enhancing the function of partially active NPC1 variants could alleviate lysosomal cholesterol buildup and reduce the downstream cellular damage associated with the disease. Unlike enzyme replacement therapies, which struggle to cross the blood-brain barrier, a small molecule like niacin, or analogs that mimic its intracellular actions, could potentially reach the central nervous system and exert its effects directly in neuronal tissue. Moreover, niacin's ability to stimulate cholesterol trafficking may also benefit other lysosomal storage conditions with overlapping lipid dysregulation, such as mucolipidosis type 4, where TRPML1 mutations similarly impair lysosomal calcium signaling and lipid export. Niacin's effect on restoring calcium dynamics in lysosomes and upregulating downstream transport proteins like NPC1 suggests that it may offer broader lysosome-targeted therapeutic value. Of course, it remains to be determined whether the pharmacological doses of niacin required for these intracellular effects are safe and tolerable in patients with lysosomal diseases, especially considering the unique sensitivities of pediatric and neurologically affected populations. Furthermore, while niacin upregulates NPC1 in macrophages, it remains to be tested whether similar effects occur in neurons, astrocytes, or other cell types most affected in NPC disease. Nevertheless, the potential is promising. Niacin not only mobilizes cholesterol from danger zones in atherosclerosis, but it may also correct trafficking defects in genetically inherited storage disorders. By enhancing NPC1 expression via LXRA and restoring lysosomal calcium flux through TRPML1, niacin could emerge as a supportive or adjunctive therapy in NPC and related diseases, bridging the gap between metabolic regulation and rare genetic disorders. In this light, niacin is not simply a cardiovascular agent, but a versatile modulator of cellular lipid handling. Its effect on NPC1 offers a hopeful frontier in treating diseases marked by lysosomal cholesterol accumulation, where few effective treatments currently exist.